In the last video, we talked about kind of the roads, right? Or how the blood is going to be serving as a road for the hormone, or how it's going to be serving as a transport corridor or as a transport vector for our hormone. This video, we're going to start talking about how we actually interpret the signal, right? How are we going to take the signal that's being released and internalize it and then respond to the stimulus, respond to that signal? And that's going to be really what hormone receptor interaction is all about. So essentially with a hormone receptor interaction, we're going to be having a specific hormonal signal that's going to be received and interpreted by a very specific target tissue. Whenever you have hormones, you're going to have target receptors as well. These target receptors are going to be responsible for taking and binding with a level of specificity these hormones that are going to be released. That could be on the outside or on the inside of the cell. It doesn't matter. There's still going to be a specific receptor that identifies a specific hormone. Once it binds, it's going to have some sort of an effect that occurs. What I want you guys to know is that hormone receptor and the interaction of the hormone with the receptor is not going to be a static structure. These hormone receptors are able to move back and forth across the cell membrane, so they're able to be recognized on the outside of the cell or on the inside of the cell. We can adjust how these hormone receptors are going to be displayed or expressed based on genetics, based on how our body is actually writing our genetic code. All these things are going to be easily manipulatable. Easy to be manipulated, I guess, is probably a better way of saying that. And it's just not a static function. We're able to actually change the ability of the hormone to exert its effect based on how many receptors our target tissue is going to be displaying. While we want to have a high level of hormone receptor specificity, meaning we want only the specific hormone to bind with the specific receptor, it doesn't always work like that. It's not always that clean. Sometimes there's molecules that can actually get in and interact with that receptor that are not the target molecule. This is actually an interesting side note because this is how a lot of drugs work. So things like ibuprofen and other sorts of chemotherapy agents and all actually work based on this level of specificity because they're able to get in and we're able to design drugs to somewhat look like a hormone or somewhat look like a signaling molecule and get in and bind with that receptor and actually affect the function of the target tissue. Realistically, we have three primary mechanisms for how we're going to exert hormone action. We're going to be able to have a direct effect on DNA. We're going to have to activate a special subset of proteins that have a special set of skills like Liam Neeson. This is going to be our second messenger system. And then we can also alter the level of membrane transport or alter the permeability of the target tissue to specific things, right? When it comes to altering specific DNA activity, this is largely going to be based on our steroid hormones or our lipid-based structures. If you remember, a cell membrane is a phospholipid bilayer. So it creates this two-level blocking system, right? Like you're going to have an outside and an inside section of the cell, right, based around the cell membrane. These lipid steroid hormones are actually able to diffuse right through that plasma membrane. And then once they get into the cell, they're able to bind with a specific receptor. And then that hormone and receptor complex, right, hormone will get through the cell, bind a receptor. That whole hormone receptor complex will actually move to the nucleus. When it gets to the nucleus of the cell, which is where all of our DNA is going to be stored, you're going to create a hormone response element. So you're actually going to have a hormone response element that's located on the DNA that is going to alter the way that we write RNA, and then we're going to have a significant effect on the amount of protein or the types of proteins that cell is creating. That means that we're going to be affecting essentially the function of that cell because of the steroid hormone. So again, let's talk through this. Lipid-based hormone, right? Steroid hormone diffuses easily through the cell. Once inside the cell, it binds to a target receptor that creates a hormone receptor complex. Inside of the nucleus is where this is going to travel to, and it's going to bind to a hormone response element. It's going to change the way that our DNA works. It's going to write a separate RNA or a new type of RNA, and that's going to affect proteins. If you really think about it, that's not exactly a super fast response, right? There's like 18 steps that have to occur, and it already seems to be that's a slow response. Think about how long it would take to change the DNA of something. It's not fast, but it's long lasting. Another major mechanism by which we signal with hormones, or we get hormones to exert their effects, is this concept that's called the second messenger system. And before we read through some of this text or talk about the text, I want you to think about second messenger system as like a game of telephone. Essentially what you have is you have a hormone binding to a receptor. That receptor is going to turn and tap on its friend's shoulder, and it's going to say, hey, I need you to do this. Then it's going to turn and tap on its friend's shoulder and say, I need you to do this. Same way that like you get orders from upper level command if you're in the military, and it kind of comes down this pipeline of, hey, I need you to do this. Hey, I need you to do this. Hey, I need you to do this. The same thing's going on with second messenger system, essentially. We have a hormone that's binding to a receptor. That receptor is going to signal for another message to be sent deep inside of the cell. So essentially, the reason why this works is certain hormones are not able to pass directly through the cell membrane like the lipid-based or the steroid-based hormones are able to do. So our peptide hormones aren't able to do this. Our protein-based or amino acid-based hormones are not able to do this like the lipids. So they actually have to bind to a specific receptor on the outside of the cell. Once it binds to the specific receptor on the outside of the cell, it will activate something inside of the cell that's called a G protein. This G protein is then going to be the link. It's going to be the individual that turns around and taps someone else on the shoulder in order to get an event to occur inside of the cell. For the purpose of this class, we're really only going to talk about two different types of second messenger systems. We're going to talk about cyclic AMP on this slide, and we're going to talk about phospholipase C on this slide. What I want you guys to see is that up here, you have a signal molecule. You have the first messenger. You have a hormone that's going to come down and bind with a specific receptor. If you notice, that hormone is not able to bind on the inside of the cell. It has to exert its effects on the outside of the cell, right? Outside, inside. When that hormone, I'm pointing with my finger, which is hilarious right now. When that hormone binds the receptor, it will actually cause an activation of this G protein right here. G protein will activate adenylyl cyclase, which will create and release this second messenger that's called cyclic AMP. And cyclic AMP is going to activate protein kinase A to exert its effects. If you notice, we have the input of ATP in order to do this, right? It's an energy-consuming step. When it comes to phospholipase C, which is the other second messenger system that we're going to talk about, you can see that out here, you're going to have the extracellular space and the intracellular space. PIP2 is going to be cleaved into an IP3 via phospholipase C. When this happens, this PIP2 is going to also release a diacylglycerol or glyceride. This diacylglyceride stays within the cell membrane and travels, and whenever this diacylglyceride, this DAG, starts to accumulate in high levels, this protein kinase C is going to grab it. How do we get that protein kinase C is where this second messenger system comes into play. So essentially, this PIP2 gets cleaved into two different things by phospholipase C. It's going to be cleaved into this IP3 and the DAG. The IP3, if you look, is actually able to serve and move into a different section of the cell. When it gets into this different section of the cell, it's going to release calcium. Calcium is going to activate protein kinase C, and you're going to get an eventual phosphorylation of substrates because of a kinase. one of the final ways that hormones are going to exert their effects is going to be by altering membrane transport, or altering the ability of molecules on the outside of the cell to get to the inside of the cell. Great example of this is going to be insulin receptor substrate, or how insulin is actually able to exert its effects. And we'll talk about that on the next slide. But essentially what happens is that you have on the outside of a cell, a hormone's going to bind a receptor. And when that hormone binds to the receptor, it's going to adjust that cell membrane in some way to either increase the permeability or to bring molecules from the outside in by adjusting the cell membrane structure or location. And that will help get whatever's on the outside of the cell inside of the cell. So it's serving as a ability to bring stuff from the outside in. So here's a great example of this. This is going to be membrane transport due to the insulin receptor substrate. So if you look, you can see that insulin binds to the insulin receptor. What I want you guys to just pay attention to is that there's a series of phosphorylation events. That's what all these P's are going to be standing for. And then these P's and this binding of insulin receptor substrate is actually able to exert several different effects, ultimately leading to the transport of glucose 4 or GLUT4 transport molecules to the cell membrane. So, a little bit cleaner, insulin binds to the receptor. This receptor causes a bunch of downstream effects that ultimately results in GLUT4 or glucose transporter 4 moving to the cell membrane. When this glucose transporter 4 or GLUT4 moves to the cell membrane, it facilitates the bringing in of glucose into the cell. And why would we bring glucose into the cell? Because we need to create energy. We need ATP.